Hello everyone. We are starting with chapter 16. So it's all about cell signaling. Cell signaling, why is it important? Let's talk about it. So, no matter what, even if you're a unicellular cell, even if you're a multicellular organism, you have to interact with the environment. If you cannot respond to what's going on outside, you are simply dead. So these unicellular organisms, they have to seek for food and dangers, and they have to sometimes seek for mates. So they have to communicate, for sure. And when we talk about the communication in such a scale, it's always chemicals. And if you're talking about the multicellular organisms, I'm sorry, they're a combination of cells, so not only one single cells, but many of them come together. Here it's a relatively simple one, but of course we are like a combination of trillions of cells, and of course they have to cooperate. If they do not cooperate, some cells will not be able to carry out their functions, which is dangerous because it will not be good for the integrity of the organism. And if some cells, for example, start to divide uncontrollably, it would be a problem again because it would create a tumor. And in our case too, cell division has to be very well controlled, and it has to communicate with lots of things around in the body, outside the body. Otherwise, it will create a tumor. And if cell division is not enough, there is not enough growth and not enough healing, so you have to control them very well. So they have to be a division of labor between cells. They have to be a good communication to when to divide, how to differentiate, etc. So all of them requires a good web of communication. So in this chapter 16, we are going to see the basis of this communication. So there are just few basic classes. Of course, these basic classes have many subclasses. There are many details, but we are not going to go in details. In chapter 16, what we are going to see is really basic. Yeah, this is, let's say, John. We are going to see John's body. So his body is made of trillions of cells, and these cells are communicating with each other, of course. Now imagine that he has some adrenal glands here, just over the kidneys, and they are, let's say, producing adrenaline. Adrenaline is the stress hormone. This adrenaline will be secreted from the adrenal glands, and it's going to be given into the bloodstream, so it's going to move to all around the body. So all of the tissues will be affected by this. So such a simple chemical, independent of where it is expressed from, is going to be distributed all around the cell. This is called endocrine signaling. It is secreted by the endocrine cells, and these molecules are called hormones. Now, I'm sure you are familiar with the hormones. Adrenaline is a very well hormone. When you are in danger, when you are excited, when you're going to do a presentation in front of a crowded people, or when you see your crush, adrenaline comes up. Your heartbeat increases, your breathing increases, etc. Your cheeks become reddish. You all know this hormone. There are other hormones in our body. Cortisol. It's also involved in some creams for the pharmaceutical purposes. There's estrogen, also called estradiol. It's actually, estrogen is a combination of hormones, and testosterone. These are the sex hormones. This is what gives the secondary characteristics of the genders. And there is the thyroid hormones, which is secreted from the thyroid glands here. Again, they are affecting several things in the body. And the thing important for us as molecular biology and genetics people is that this adrenaline or the hormone is something tiny, something very small, and it's going to affect various tissues. And each of the tissues that is going to be affected is going to be affected differently. Think about your cheeks after adrenaline. They are going to get reddish. think about your heart, it's going to increase the pace. Think about the muscle, they will start to work. So each tissue is going to respond in a different way. So what makes different tissues respond differently? That's the question we are going to answer. And second, there are paracrine signaling. Now, paracrine signals, they don't affect all the body, but they affect a region. Now, let's say around here, a bee stitched this guy, John. Now, after the bee, it starts to secrete a local mediator. Let's say this time histamine. Oh, I'm sorry. Okay, histamine is expressed, and not only around the point that the needle of the wasp or bee inserted, but all around the tissue is now itchy, and it starts to be reddish because histamine is a local mediator, and it's going to mediate the cells around. It's going to affect what is going on around. And it's going to, maybe it may start inflammation. Inflammation is actually the first level of defense of the immune system. And it's not going to affect the entire body, but a good amount of cells in a local region will be affected. The histamine is a very well-known example, especially when you're itching, you feel that deeply. There are other examples, growth factors. Now, when the tissue is going to grow, the decision is given both centrally and locally, and these are the growth factors that are expressed locally. EGF is the epidermal growth factor, PDGF is the platelet-derived growth factor, NGF is the nerve growth factor. So this is required for the division of the neurons. I'm sorry. And there are some other things that we are going to see. For example, nitric oxide. Although it is not well known, it's actually a local mediator, and it changes the behavior of cells locally in a tissue. Okay, so in compared to hormones, which affects the entire body, paracrine signaling affects a smaller region. And if we talk about the neural signaling, it is going to affect just two tissues. Now, let's say there is a neural signaling, and here it starts, and it affects here. Now, although it is going a very long distance, the signaling is very direct. So the cell here is affecting the cell here. So this is a neural cell, and this is most possibly a muscle cell, if it's a motor neuron. So it is traveling a very long distance. This arm is giant in terms of cell signaling, and it's not going to affect the finger here. Oh, sorry. It's not going to affect the finger here. It's not going to affect the knee here or the cell here. Only and only this cell will be affected because it's a direct connection. Okay. This is a synaptic interaction. So the two cells are interacting again with chemicals. And here the case of hormones and paracrine cells, they were also chemicals. In the case of synaptic transmission, it's again chemical signaling. This time we don't call them endocrine or paracrine. These are the neurotransmitter cells. And it is not diffused into a long place. It is just diffused between the presynaptic cells and the postsynaptic cells in a very narrow place. And finally, there is the contact-dependent interaction. Now, it is very, very direct, and it's not even traveling long distance. Now, think about two cells in John's body, just two cells. These two cells, maybe I'm going to even make it bigger. These two cells are going to interact directly. No other cells around them will be affected. So this is one-to-one interaction of these two cells, and a good example of this is Notch delta. And these one-to-one interaction frequently take place during development, embryonic development. So after John is an adult, maybe you're not going to see that anymore, but when John is an embryo, John was an embryo, we used to see it all through the neural development. So here is an introduction of a large class of cells signaling. And the next question is, how do the cell respond? Now, we have a cell. Okay. Now, this cell is, of course, a bag of, let's say, chemicals. And the chemicals, of course, there are many things around, and it is looking for interaction, it is looking for information. Any kind of information the cell is going to receive is in the form of chemicals. Okay, even if it's a neural cell, the information travels within the neuron, within the axon as electricity, but from one cell to the other cell, it is nothing but chemicals. So the signal is going to come from somewhere far away. This somewhere far away can be endocrine or paracrine or nerve terminal, and even it can be contact dependent, but at the end it is going to come to the cell. Now once it comes to the cell is going to respond, but of course to respond it needs a receptor. So having a signal is not enough. Having a signal means nothing unless you have the proper receptor. So if somebody has no receptor, and if you're giving the signal, it's like talking to somebody deaf or showing colors to somebody blind, because there is simply no receptor and that person is not going to recognize what is going on. So for every signal we have receptors. We have to have receptors, otherwise we cannot understand it. Now, a good content of this chapter 16 is the difference in between receptors, and we know that there are different receptors in different kind of cells. Now, let's first check the heart pacemaker cells and salivary gland cells, and this time the signal is the same. Acetylcholine, a very simple molecule, a very simple communication manner. Now, this acetylcholine is going to be received by this transmembrane protein in the heart pacemaker cells. And once it is received, it is going to decrease the heart rate. So instead of beating very strong, it is going to be a little bit slowly, it is going to calm down, and this is the effect on the heart. Now, let's check the salivary gland cells. The receptor is exactly the same. It's the same transmembrane protein. It's the 7th transmembrane protein. It's actually a GPCR. And once the salivary gland receives it, it's actually going to do exocytosis. And remember this chapter 15 content. This exocytosis is not constitutive. This is a regulated exocytosis. Once the signal comes, these vesicles, these vesicles, they are going to fuse with the membrane. Anyway, they are ready to fuse. The snare proteins are waiting for that. And once they are there, the salivary gland will secrete the salivary material. So the very same receptor, but the action is different because the intracellular composition of these two cells are different. You can think it like a black box. Okay, I'm going to come to this black box analogy later. And now think about this cell. This is a muscle cell. Here, even the receptor, even the transmembrane protein is different. Look, it doesn't look like anything like this. So once it receives the signal, it's going to react very differently. It's going to contract. So the same acetylcholine signal, the reaction can be very different because each time a cell receives a signal, a computation is going on here. And this computation is totally molecular. So there is a series of molecular interactions. Some molecules are affecting the other molecules. At the end, they come up with a decision and they cause a behavior. This behavior can be contraction, can be secretion, can be decreasing rate, increasing rate, whatever. But it's a specific thing that is a result of the combination in the cell. Now, Of course, the reaction can be different, and what I didn't tell in the previous one is that one cell doesn't receive one single signal, it's actually receiving several signals at the same time. This is very normal, just like that you are hearing and seeing several things at the same time. For example, you are hearing me and you are following the pen and you are checking the figures at the same time. You're getting many signals, many stimulus in the meantime. You're combining them and you're coming up with a decision. For example, in this case, these three signals, when they combine, they mean survive. So, of course, survival is not a default. The default is the death. The default is the suicide. So this is the same for many multicellular cells. For many multicellular organisms, this is a prevention against cancer. When the cell doesn't receive any signal, the default thing that it is going to do is to kill itself. In order for a cell to survive, every day you have to tell the cell, don't die today. And for that, you have to give very specific signals, which we are going to study. If the cell doesn't receive the combination of don't die today, survival signals, it's just going to suicide. Okay, and there can be other signals, and here, see, it's a combination of the survival signals plus some other signals. It's going to divide. It's going to go under mitosis. Of course, mitosis is not the result of one single signal, it's a combination of several signals. And think about it as the survival signals plus some other signals. The cell is going to change characteristics and it's going to differentiate. So it is all about the interaction of chemicals, it's all about the chemical signals that the cell has, nothing else. And of course, there's a matter of speed. Some reactions are very fast. Now, think about that. This signal, this extracellular signal, comes to the cell. It is received by the cell. Now, if there is a protein which is already there and which is already, let's make it colorful, which is already waiting for a signal, this protein is going to react very fast because anyway it is there. It doesn't have to be transcribed, translated. It's already waiting for the reaction. And what usually happens is that once this protein is induced, the post-translational modifications change, and with this post-translational modifications, it reacts within seconds or within milliseconds. This is, for example, what happens in the nerve terminals. Once the signal comes, the snare proteins, the calcium activated proteins change shape, and once they change shape, the signal is relieved immediately. But in some other cases, the protein is not there, so instead the signal has to travel all the way to the nucleus. It finds the DNA, the responsible gene. This gene is first transcribed and it's made to RNA, which takes some hours, and then it's modified. You remember there's the 5' capping, 3' poly tailing, and then the introns are spliced out, and there's a quality control after it is regulated, and it's so that it can pass through the nuclear pore complex, and then here it is translated, which takes some more time, and then the protein is going under quality control, and then if everything is okay, it's going to function. So the functional protein will be here. So it can take few minutes if it's really optimal, but mostly it takes hours. So this is a slow reaction, and this is a fast reaction, and this is a slow reaction. So a cell is going to respond what is going on around, doesn't mean that it's going to respond in seconds. Think about the changes in your body when you're adolescent, when you're a teenager. And it took days, months, or years, which is very normal. Or think about getting excited or getting nervous. It responds within minutes or seconds, which is also very normal because these are two different circumstances requiring two different